So I'm going to be talking about biogeochemical processes on glacier and ice sheet surfaces. So when we talk about glaciers and ice sheets, about 10% of the world's entire landmass is covered by them, and they store about 75% of all the fresh water on Earth. And polar regions play a very vital role in Earth system processes. The cryosphere, the hydrosphere, and the atmosphere, together with all the biological communities which live in and around the polar regions, are a closely interactive component of a very complex climate system, which has both regional as well as global influences. And for a very long time, glaciers were thought to be biologically inert. And in recent years, there has been a shift in the way we regard the glacial environment. So the top few centimeters of the glacier surface are like a frozen rainforest. It's not really the empty white wasteland that people imagine it to be. So if you look at a microscopic scale, there is an abundance of life. And microbes living here have to overcome combined stresses of low temperature, desiccation, fluctuating light levels, from continuous light in the polar summer to continuous dark in the winter. And they also have to deal with elevated levels of UV radiation. And of course, there's limited availability of nutrients. But these microbes have found a way to overcome these stresses, and they are active at very low temperatures. And although hundreds and thousands of kilometers away from where we live, these organisms have a huge significance for everyone on Earth because they play key roles in the melting of glaciers and ice sheets, which I will explain in the coming slides. They affect carbon dioxide concentrations in the atmosphere. They contribute to cycling of key elements and nutrients, and so on. And these organisms could also teach us about the potential of life elsewhere on our solar system. Now, when we look at the glacier from high above, the ice sheet appears to be quite a featureless wasteland. But often the surface is riddled with thousands of holes like these. And these holes are called cryoconite holes, and they look very simple, but they're actually a main habitat on the ice surface and are home to a very rich ecosystem of organisms. And they're actually one of the most biodiverse microbial habitat on the Earth's ice. And basically these holes form when you have dust and debris on the glacial surface, when they get tangled up by long thread-like photosynthetic bacteria called cyanobacteria. Now, these cyanobacteria, as they grow, they start producing, sticky polymeric substances that act as biological glues, and it bundles all these materials together into very stable granules. And this biological bundling and binding sort of creates a microhabitat for other organisms, especially those who can feed on the molecules produced by these photosynthetic cyanobacteria. So, as you can see, as the granules grow, they become dark. And since they're dark, they absorb more of the solar energy, and then they melt into they cause the melting of the ice around it and causing holes to form on the ice surface with the granules sitting on the hole floor. And the holes provide protection from weather and intense sunlight and also provide liquid water for the microbes. And so we can say that the cyanobacteria therefore sculpt the ice surface and engineer very comfortable and stable habitat where diverse microbial communities, can thrive on the glacial surface. Another habitat on the ice surface is the small, thin liquid water films between the crystals of ice. So basically, when snow crystals transform into ice granules, there's a thin liquid layer forms on the surface of the snow, on the surface of the snow grain, and this is enriched with ions and nutrients. And there you have bacteria and algae. Now, these ice algae, they are very dark because they produce carotenoid pigments as a protective mechanism against UV radiation. And these pigments darken the algae and therefore the ice which contains them, especially if the algae are present in very large numbers. And the color of the ice is really important because color would determine how much solar energy is absorbed compared to how much is reflected away. And the amount absorbed will determine how much energy is actually available to melt the ice. So explorers since long, have tried to draw the attention of the public to the importance of life in these, inhospitable regions. Even as far back as the 1870s, a polar explorer called Adolf Nordenskjold noted that the ice on his expeditions to Greenland were often brown, gray, or black. And after checking some samples under a microscope, he discovered traces of bacteria and algae in them. And he noted down in his diary that microscopic life was the greatest enemy of the mass of ice. And until recently, Nordenskjold's observations of life on ice have remained obscure footnotes in the history of polar exploration. But in the 21st century, as climate science has become increasingly urgent, Nordenskjold's work has gained new significance because scientists have now confirmed the presence of microbial life on the surface of the Greenland ice sheet and elsewhere, and they are now actually attempting to quantify their ice darkening effect. Now, this is a photograph of an area of the ice sheet, and you can see that it is creviced and pot-marked with deposits of cryopyite. And as you can see, these cryopytes turn the snow dark. And dark snow absorbs more sunlight and accelerates the melting process. So this is about, so there is this new concept emerging about how biological forces influence the albedo of the Earth's glacier and ice sheet. So albedo is simply the reflectance of solar energy back to the atmosphere. So if something has high albedo, it is reflecting most of the sun's energy back so the surface can remain cool. If, on the other hand, something has low albedo, it is absorbing most of the solar radiation and it is becoming warmer. Now, in the context of ice, it's really bad news to have low albedo because it means that solar energy is being pulled into the ice, making it to melt faster. And as microbes hasten this melting process, there is actually a positive feedback because the more melt you generate, the more microbes can feed on that are released within the melt water, and then you get more microbial growth. So this is actually a vicious circle of how microbial growth and melt are driving each other. So biological processes are very closely driving positive feedbacks in ice melt, and that is a scary thing because current projections of how glaciers and ice sheets respond to climatic movement don't really include this biological albedo feedback. And the first experimental evidence that microbial activity can significantly impact the reflectivity of the ice surface was done in 2016 by Musilova et al. And what they did was an experiment was conducted to investigate the darkening of these cryopyite debris as a result of microbial activity. And cryopyite debris, which basically comprised of 10% natural cryopyite mixed with 95% of cryopyite furnace at 550 degrees to remove all the organic matter. This cryopyite was exposed to simulated Greenlandic summer conditions in terms of temperature, lighting, nutrient availability. And this cryopyite mixture basically simulated the early stages of cryopyite hole development, where the debris is mostly inorganic and it can later on get colonized by local microbial communities. So samples were kept under light, as shown in yellow, and in dark conditions, shown in green. And amendments of nitrogen, phosphorus, and carbon were carried out, that is NPC, at ice meltwater concentrations. So figure A shows the organic carbon accumulated over one simulated summer season, and B shows organic carbon accumulated over 3 simulated summer seasons. And as you can see, the light samples, the yellow, accumulated significantly more organic carbon compared to dark samples. And then correspondingly surface reflection after one simulated summer season, that is C, and after 3 simulated summer seasons, that is B, showed that the accumulated organic carbon over a period of melt season actually darkened the debris and reduced the reflectivity of the snow from 31 to 15%, generating a very strongly absorbing surface. And this reflectivity reduced still further over a period of 3 melt seasons. And these experiments were basically the first to quantify the microbial-induced melting of the ice sheet. And the results from this study were then used in a regional climate model to project future microbially mediated increases in ice melt. So figure A shows the present biologically induced increase in melt rate in millimeters per year, and B and C show future biologically induced increase in melt using two different model parameters. And the more of the blue and the red and the yellow basically indicates that there's more biologically induced melting of the ice sheet. And the study provides the first order estimates, which indicate that the present microbially mediated melting is about 17.3 gigatons per year, and it can go up to 85 gigatons per year by 2100. Thus, biological impact on albedo plays a very important role in modulating the mass loss from the glacial surfaces and must be included in albedo models if we want to adequately capture how glaciers evolve in a changing climate. And also it is believed that as the climate warms, the melt seasons will be extended, leading to increased biological activity, and then this would further contribute to the darkening of the glaciers and ice sheets. Now, microbes living in supraglacial environments also play another important role, and that is of the cycling of carbon and nutrients, especially nitrogen. The inventory of nitrogen compounds in the polar ice sheet is approximately about 260 teragram of nitrogen, and largely dominated by nitrate in the much, especially in the Antarctic ice sheet. And active biological nitrogen cycling occurs on the snow surface, wherein compounds like ammonium, nitrate, nitrite, chiefly supplied by snow precipitation and wind, are cycled through different pathways and into organic nitrogen compounds. And these different forms of nitrogen are then available for other organisms to use. And here the thickness of the arrow just represents the number of microbial sequences associated with each process. And similarly, active nitrogen cycling has also been shown to occur in pyritinite hole granules. And ice sheets also matter for the global carbon cycle because cyanobacteria and algae on the ice surface capture carbon dioxide from the atmosphere and convert it into organic matter. Other bacteria then break down this organic matter together with organic matter from other sources and generate carbon dioxide that is released back into the atmosphere. So the balance between these two processes will determine whether a glacier is a net sink or a source of carbon dioxide. So this figure shows the changes in carbon produced, the first figure, and consumed over one ablation season or over a melt season in different glacier habitats like clean ice, dirty ice. Dirty ice basically means ice containing a lot of dust particles, and cryogenite water and cryogenite sediment. And all the different glacier habitat types studied were active and net autotrophic systems, meaning they produce significantly more organic carbon through primary production than is being consumed by respiration. So overall, it shows that microbial communities drive the organic carbon production and recycling on the glacier surface. And to date, about 3 attempts have been made to model these carbon fluxes in the supraglacial environment, and you can see that how ice sheets have a huge contribution to this global carbon cycle. And what we have tried to do is basically try to understand at the molecular level what this carbon on the ice surface is composed of. So our studies, including few others, have shown that there are three principal types of organic substrates available on the ice surface. One is organic carbon freshly produced by resident microbes, that's like lipids, proteins, carbohydrates, and these are readily available for other microbes to use. And you also have organic carbon transported from far away, like lignins, tannins, or black carbon particles because of soot that is deposited on the ice surface. And these can also be utilized by some microbes. And you also have, in addition, organic carbon that is coming from local sources, like exposed soils, or marine sources, etc. And what we attempted to do is we tried to look at what happens to this carbon once it is deposited on the ice surface. So we carried out in situ field experiments in Antarctica, combined with basically mass spectrometry, to show for the first time at a molecular level or at a compound specific level the bioavailability of distinct molecular groups. So the green circles basically represent organic molecules detected on the snow surface. And then this snowpack was then subjected to different treatments, light only, which means that basically all the microbes in the snowpack were killed and the snow was exposed to light. Microbe only, where the microbes were retained, but the snow was shaded to prevent light penetration. And then the light plus microbes, where the microbes were retained and the snow was exposed to light, because we wanted to see what is the impact of solar energy plus microbial activity on the organic matter composition over a period of time. And when we looked at the snow again after 35 days, what we found that in each of these treatments, there were massive changes in the organic matter composition. The black circles basically denote the original organic molecules present in the sample, and the red circles indicate the new ones that were produced. So these photochemical and microbial processes resulted in massive changes in the organic matter composition, where several new molecules were formed. And these new organic compounds resulting from these processes had very different properties when compared to the original sample. And we also saw shifts in nitrogen, sulfur, and phosphorus content of this organic matter. And these processes were also important, interestingly, in transforming compounds that we traditionally consider to be refractory, like black carbon, dissolved black carbon, and lignin and tannin molecules, etc. And thus, photochemical and microbial processes occurring individually or in combination played a very crucial role in determining the composition of the bulk organic matter in these environments. And our studies and that of others show that about 22 to 94% of the supraglacial carbon is actually bioavailable. And attempts have been made to quantify the organic carbon in glaciers around the world. And these glaciers have been shown to be, contain huge reserves of organic carbon. And over the past decade, we all know that the Antarctic ice sheets and other glaciers have been disintegrating at a very alarming rate. And it is estimated that the annual release of glacier organic carbon via ice melt or calving to these downstream freshwater marine ecosystems is about 3 teragrams. That is, 10 to the power of 12 grams. And these losses are expected to accelerate to about roughly 15 teragrams of glacial organic carbon by 2050 due to climate change. And to put this in perspective, this would be equivalent to about half of the annual flux of organic carbon from the Amazon. So, glacial organic carbon entering aquatic ecosystems is likely to simulate microbial metabolism and therefore ultimately contribute to carbon dioxide outgassing to the atmosphere. And it can also impact marine food webs because, like I showed you, much of this organic carbon is derived and available from other organisms. So it is quite clear that glaciers represent a substantial reservoir of organic carbon. And as a result, the loss of glacier mass worldwide, along with the corresponding release of carbon, will affect marine ecosystems, particularly those that are surrounding the major ice sheets. So, we now understand the major biological processes occurring on the surface of glaciers on a conceptual basis. That we know that microbes can aggregate supraglacial debris and that they can produce dark compounds, which can decrease the surface albedo and thus promote ice melt. We also know that supraglacial microbes can sequester carbon dioxide from the atmosphere and then break down this carbon together with carbon from other sources for its use. And microbially modified organic carbon and nutrients can also be exported to downstream ecosystems, potentially with impacts for marine food webs and biogeochemical processes. But there are still many blind spots and missing links in our understanding of these biological processes on glacier surfaces, because the influence of microbes on surface albedo has not yet been assessed in a really quantitative way, nor has the export of the supraglacial organic carbon, or its consequences for downstream carbon cycling. Another gap is the absence of quantitative and often qualitative data on the inputs of microbial cells or organic substrate and other nutrients to supraglacial environments. And finally, results of most studies that are dedicated to supraglacial biology come from accessible sites, and data from remote glaciers, particularly southern hemisphere, are lacking. Therefore, we have little knowledge of large-scale variations in biological processes on glacial surfaces. And integrated studies of these whole glacial systems, which take into account the carbon, nutrient, energy fluxes, will be necessary for a more quantitative understanding of climate cryosphere interactions. So, to end with, I believe that the study of life on the Earth's ice is deeply interdisciplinary. And ultimately, I think it requires all of us to recognize, as Nordenskjöld did, the intricate bridges that join the very big and the very small. Thank you.